So what do I mean when I say something is an activated carrier? Well, these are molecules that carry functional groups that are eventually transferred during metabolic reactions. It can be useful to know which functional group each activated carrier is associated with, since this can help determine the output of each metabolic reaction. However, just note that the exact structure is not going to be important for the USMLE. We'll start off with our first one, adenosine triphosphate, which we already talked about. It carries three phosphoryl groups. It can use these high energy phosphate bonds to further phosphorylate proteins and thus drive reactions. We also have NADH shown here, as well as NADPH and FADH2. Now, all three of these are going to carry electrons. These electrons can then be traded in later on down the road to make some ATP molecules. Coenzyme A, shown here, and lipoamide carry acyl groups. Biotin is going to carry carboxyl groups, so carboxyl groups, and then tetrahydrofolic acid is going to carry single carbon groups. S-adenosylmethionine is also known as SAM, and this is going to carry methyl groups. So our methyl group is going to look kind of like this. And then lastly, we've got thiamine pyrophosphate, which is actually the active form of vitamin B1. This is going to carry aldehyde groups. So that's our aldehyde group carrier. Thinking back just a little bit, in the biochemistry nutrition section, he talked about the most common electron acceptors, which are the nicotinamides, our NAD+, and our NADP+, as well as the flavin nucleotide FAD. So let's go ahead and think back to that biochemistry nutrition section. Do you remember what vitamin is our FAD derived from? Well, our FAD is derived from riboflavin, or vitamin B2. So on the other hand, our NAD and our NADP are both derived from niacin, or vitamin B3. So all three of these, we're talking about our universal electron acceptors, all three of these molecules here are going to accept electrons using various redox reactions. And then they're going to transfer those electrons to other molecules, say the proteins in the electron transport chain. Our NAD is going to be used mainly in catabolic processes that break down organic molecules. So here we have a big organic molecule. We're going to take an NAD, feed it into our system. We're going to run that system and we're going to get some smaller molecules. We're breaking down that big organic molecule, and then in the process we're transferring electrons and we're going to get some NADHs out of that. So if NAD is primarily used in catabolic processes, what is NADPH used for? Well, it'll make sense that it's used in anabolic reactions such as steroid synthesis and fatty acid synthesis. So we take a molecule of NADPH, feed it into our system. We're taking these smaller building blocks now and going to build a big molecule. So let's go ahead and run our reaction, and we get a big molecule, and in the process we get NADP+. So NADPHs can be created using the hexose monophosphate shunt, aka the HMP shunt, which we'll cover soon. It's also used in other places, like during the respiratory burst to create oxygen radicals to damage bacteria, in the cytochrome P450 system, which we'll cover in the pharmacology unit, and as well in the glutathione reductase pathway to reduce glutathione. So multiple uses for our electron acceptors. Now we can really jump into the meat of the biochemistry unit. And we'll talk about the first step of glycolysis, which, remember, glycolysis is the metabolic process that's going to break down our glucose and form ATP. So do you remember, what is the function of a kinase enzyme? Well, thinking back to our discussion about terminology, the kinases are going to transfer a high energy phosphate group from ATP to a substrate. So we're going to be dealing with our kinases. Remember I said that if you know the name, it'll give a clue to what the function is. So here we see a cell outlined in red, and we'll have a molecule of glucose outside of that cell in this orange box. It's going to be free to enter the cell and then leave the cell as it pleases in its current form. However, once the glucose enters a cell, it's going to meet with an enzyme called hexokinase or glucokinase which is going to add a phosphate group, a blue circle in this case, onto this glucose molecule. So thus, the first step of glycolysis is the phosphorylation of glucose to create a glucose 6-phosphate by using 1 ATP. We took an ATP, grabbed a phosphate off of it, added it to the glucose at the 6 carbon position, and now we have a glucose 6-phosphate. Why is a phosphate added to our glucose? Well, in addition to prepping glucose for the next step of glycolysis, this also gives glucose an overall negative charge, which is going to prevent it from passing through the plasma membrane, effectively trapping that glucose in the cell. This reaction can be catalyzed by either the hexokinase enzyme or the glucokinase enzyme. The differences between these two are their locations and their enzyme kinetics. We'll talk more about enzyme kinetics in the pharmacology section, especially the tricky concepts of Km and Vmax, which you can see kind of here in this graph. We'll talk more about all this later on in the pharmacology section. For now, however, I'm going to focus on the kinetics of these two specific enzymes, our hexokinase and our glucokinase, in the next slide. So let's talk about hexokinase first. Hexokinase is found in almost every cell type of the body, with two exceptions. It's not going to be found in liver cells, and it's also not going to be found in pancreatic beta cells. So hexokinase has a high affinity for glucose, but it has a very low reaction rate. So let's go ahead and see what that is visually. We have a very slow, methodical phosphorylation of glucose here. Because remember, it's our kinase that's going to add those phosphate groups at a nice, slow, predictable rate. So what does it mean when you have a high affinity and a low Vmax? Well, it means that the concentration of glucose needed for this enzyme to be used is relatively low since it has a very high affinity. It'll grab onto the glucose that it sees. However, the speed at which the reaction occurs is also pretty low due to its low Vmax. Hexokinase is not going to be affected by insulin, but it does receive some negative feedback from high amounts of glucose 6-phosphate. So it will have glucose 6-phosphate inhibition. Remember, glucose 6-phosphate is the product of this enzyme. It's going to take a glucose, attach a phosphate to it, and now it's glucose 6-phosphate. So if you have high levels of glucose 6-phosphate, you're going to get some inhibition. So basically, when the cell has low glucose 6-phosphate levels, this hexokinase enzyme will catalyze any glucose available into glucose 6-phosphate at a nice, slow, predictable low rate. However, if there's a large amount of glucose in the blood, such as after a meal, hexokinase won't be able to keep up since it has a very low speed of reaction. And now we can jump to our second enzyme, the glucokinase. High glucose loads, like after a meal, is where our enzyme glucokinase comes into play. Where is glucokinase found in the body? Well, glucokinase is only found in the liver and beta cells of the pancreas. Thinking back to our last slide, I said that hexokinase was found everywhere except for these two spots. So glucokinase is going to be found here. Glucokinase has a very low affinity for glucose, but a very high capacity or reaction rate. That means that it's not going to be active unless if you have a lot of glucose in the bloodstream. But if you do have a lot of glucose, it can act very quickly to phosphorylate it. Notice how this was much quicker than our hexokinase enzyme. It also has no feedback inhibition from our glucose 6-phosphate. So as long as there's a lot of glucose in that bloodstream, our glucokinase can keep on phosphorylating it. You can also think of it as being kind of a buffer system because after you eat a large meal, for example, so we eat a large meal, what's going to happen? Well, you're going to get a lot of glucose in the bloodstream. If you have a lot of glucose in the bloodstream, it's going to activate beta cells in the pancreas, which are going to release a lot of insulin. And here I can just kind of draw an arrow here. Our insulin then is going to affect our glucokinase enzyme. And what it's going to do is it's going to activate it. It's going to tell that glucokinase enzyme, you need to start phosphorylating all these free glucose molecules in the bloodstream. Now you can remember that glucokinase is found in the liver since both liver and gluco have 5 letters each. So gluco and liver, 5 letters each. Just a little trick for test day if you get tripped up. So if all else fails on test day, what is another way to think of hexokinase and glucokinase? Well, I like to think of these two enzymes kind of like the tortoise and the hare. Hexokinase is our tortoise because it acts at a nice slow, steady pace phosphorylating our glucose, while glucokinase, on the other hand, is the hair, which is going to speed up and slow down depending on the amount of glucose actually present in the bloodstream. Before moving on, I also want to touch upon the concept of maturity onset diabetes of the young, or MODY for short. You'll learn more about the disease diabetes as a whole in the endocrine section, but in a nutshell, diabetes is going to result in an increase in glucose in our bloodstream. Now, there are many different forms of diabetes out there. So, for example, there's going to be type 1 diabetes, there's going to be type 2 diabetes, and then we also have our maturity onset diabetes of the young, MODY. We'll touch upon MODY here in the biochemistry metabolism section. Maturity onset diabetes of the young is going to be a monogenic disease, meaning there is usually only a mutation in one gene causing the diabetes, versus these other forms of diabetes which have multiple causes, or a polygenic origin. So these are usually polygenic, while our MODY is monogenic. Easy way to remember that, MM for testing. So one of the mutations in this MODY can involve the glucokinase gene. Remember our glucokinase enzyme which we're talking about now. So this mutation will functionally alter our glucokinase enzyme, producing a form of diabetes. Now, why is this? Well, let's go ahead and try and draw this out. I'm going to try and reproduce the cell, and we have a glucokinase enzyme inside that cell. We have a bunch of free glucoses in our bloodstream outside of the cell, and we also have insulin. Now, insulin normally is going to activate our glucokinase enzyme, because we would want to bring all of that glucose inside of our cell, trapping it. So that's normal. So if a point mutation causes this glucokinase enzyme to malfunction, so our insulin is not going to activate that glucokinase like it normally would. We have that mutation. So this is going to result in an increase in glucose outside of the cell in our bloodstream. So why is this? Well, it's because our glucose normally is going to enter our cell. It'll become phosphorylated by our kinase, which is going to trap it inside the cell. However, now that our glucokinase is not going to be working as efficiently, this glucose can now leave the cell as well, so more glucose will be in the bloodstream. It's also important to note that hexokinase has not been associated with MODY. This is particularly only with glucokinase, the MODY. So we have a 65-year-old man with type 2 diabetes, and he injects himself with insulin before his lunch. How does insulin affect the action of hexokinase and glucokinase? Well, hopefully you recognize that insulin induces the enzyme glucokinase, but it does not affect the enzyme hexokinase. This is because hexokinase is always active, while glucokinase, on the other hand, must be stimulated by insulin when glucose levels are high, and this creates more glucose 6-phosphate to trap that glucose inside the cell.